So, when your body is actively absorbing a meal compared to when your body has completed the absorption of a meal and for several hours afterwards, your body does different things in order to make sure that you maintain blood glucose homeostasis. And so, these different periods of time are called the absorptive and post-absorptive states. Pretty soon after you consume a meal, your nutrients will begin to enter your blood. And there's two metabolic hallmarks with this. First is you have the oxidation of glucose. You're going to be making ATP in your body cells so you can replenish your ATP supply. And inevitably, you'll have some excess. And these excess fuel molecules will be stored in hepatocytes in your liver, adipocytes in your fat tissue, and your skeletal muscle cells. And during this absorptive state, when you're actively absorbing a meal, those nutrients are entering your blood after being digested, the pancreatic beta cells are going to release the hormone insulin. Insulin helps to move glucose and amino acids into your body cells so that they can use it to make ATP. So here are some of the pathways during the absorptive state. So here is your GI tract, and you are absorbing your broken down components of your meal. So you have amino acids that were from the protein you ate, glucose from the carbohydrates you ate, and you have triglycerides that are in the chylomicrons being transported in the blood from the lipids that you ate. This glucose, some of this is going to be going to your tissues, and you'll oxidize it to create ATP. So glycolysis, for example. Insulin is going to help facilitate this. It acts as kind of a ferry in order to get this glucose into your body cells so that you can create ATP. Excess glucose is also going to be taken to your liver. And again, insulin is the hormone that's going to mediate this. There are a couple of different possible pathways for glucose. It can be stored long-term as glycogen, which is your storage carbohydrate. It can be transformed into glyceraldehyde 3-phosphate or fatty acids that ultimately are turned into triglycerides, which is a lipid. So you can turn carbohydrate into lipid thanks to your liver. You can also create some ATP in your liver. The triglycerides are going to be transported to your adipose tissue where they can be stored. And this is thanks to your VLDLs, your very low density lipoproteins. They transport these triglycerides from your liver to your adipose tissue for storage. Some of this glucose also goes directly to your adipose tissue, where you see these same processes. You can transform that glucose into glyceraldehyde 3-phosphate or fatty acid and create triglycerides here in the adipose tissue. So same idea as what we saw in the liver. Another pathway for glucose is it can be stored in your skeletal muscle as glycogen. Triglycerides that you've absorbed from your gastrointestinal tract can also be taken directly to your adipose tissue. Amino acids can be converted to keto acids, which then can go into creation of ATP. It can go into the creation of fatty acids and then be stored as triglycerides. So in addition to glucose being converted to lipids into triglycerides, you can also convert amino acids into triglycerides for long-term storage in adipose tissue. And then you can also have some of these amino acids that can be put together to create protein. So there's your protein anabolism, creating proteins from individual amino acids. Some of these proteins are also taken to your skeletal muscle. Here's an overview of how insulin is going to help regulate your blood glucose. So if your blood glucose rises, which is what's going to happen as you're absorbing a meal, right? As you have digested your carbohydrates, your lipids, your protein, those nutrients are going to be absorbed into your blood. And so as you're absorbing a meal, your blood glucose will rise, and this will stimulate your beta cells of your pancreas to secrete insulin. So you have an increase in insulin. Insulin will target tissue cells, and it helps with a couple of things. It helps transport amino acids and also glucose into your tissue cells. And so by moving those amino acids into your cells, you increase protein synthesis in that cell. And then for the glucose, some of that will be used as storage. If you have excess glucose, you can turn it into fatty acids and glycerol. Those are the components to create a triglyceride. You can store that glucose as glycogen, but this will be your excess glucose. So whatever ATP you need, you'll make through cellular respiration. And so you'll break down that glucose, use glycolysis, and all of those processes of cellular respiration to create ATP. So your cells create the ATP that they need from the glucose, and then excess will be stored as glycogen or as triglycerides. When it's been about 4 hours since you have completed absorption in the small intestine, your blood glucose levels will start to fall because it's been a while since you've eaten, it's been a while since you have put new nutrients into your blood. And it's important for your body to maintain blood glucose homeostasis. And so you need to tap into some of those reserves, some of that storage that you created in the absorptive state in order to maintain your blood glucose levels. And glucagon is the hormone from the pancreas that will help to mediate this. And so instead of shuffling glucose into your cells to create ATP and putting all the rest into storage, now you're going to start taking out some of that storage and creating glucose from other ways. So glucose production, you can break down glycogen into its individual glucose monomers. You can break apart lipids during lipolysis. You can create glucose from non-carbohydrate sources through gluconeogenesis, and you can use that, you can create glucose from lactic acid, amino acids, or triglycerides. You're also going to conserve the glucose that you have, and so you can oxidize fatty acids, lactic acid, amino acids, ketone bodies even, and you can break down muscle glycogen. So you're going to conserve the glucose that you do have by breaking down other non-glucose sources. Especially for your brain is very picky. It's necessary for your brain to consume glucose in order to create its energy. Most of the rest of your body can consume many other types of nutrient molecules, fatty acids, lactic acid, triglycerides, but your brain is picky. And so you want to, if your blood sugar starts to fall, you want to conserve what you do have and make sure that your brain has enough glucose. So that's the glucose sparing effect. It's a little bit different than just glucose conservation, but that's one of the ideas behind why you would want to conserve your glucose. Here are the main pathways during the post-absorptive state. So instead of focusing on the gastrointestinal tract, now we're focusing on the liver, because the liver is the organ that helped you put all of these energy molecules into storage in the first place, right? So the liver is going to take its glycogen, break it down, and put it into glucose. Glucose can then go into the blood. Your adipose tissue can break apart its triglycerides into fatty acids and glycerol and send that into the blood. When it reaches the liver, you can transform the glycerol and fatty acids back into glucose, and that glucose heads into your blood. Lactic acid that's available can also be converted to glucose, and you can take some of the proteins, and this is really just if you're fasting for a long time or if you are undergoing starvation, you can break apart your proteins into individual amino acids, and then the amino acids can be converted to keto acids and then to glucose. Again, this is a last-ditch effort to make sure your body has energy. Generally, you will not be breaking down your muscle proteins unless you're starving. Fatty acids can be taken from the blood to produce ATP. So you see that over here. The heart can use fatty acids for energy to create ATP. And then any other tissues, you can break down proteins. into amino acids if you need to. Fatty acids can be used to produce ATP as well. Lactic acid can be used to produce ATP. Amino acids can be used to produce ATP. Fatty acids can be converted to ketone bodies, and you produce ATP from those as well. In your brain really prefers glucose as its primary ATP source. It doesn't really like to consume other energy types. It can consume ketone bodies, but really glucose is the preferred method by your nervous tissue in order to produce ATP. All the rest of your body can use all of these other energy sources, fatty acids, lactic acid, ketone bodies. You can transform triglycerides or protein if you need to for anything you want, but your brain has to use glucose. The way that glycogen gets transformed is basically by getting broken down into glucose, and then glucose can go through glycolysis to be turned into 2-pyruvic acid, release a little bit of ATP in the process, and then pyruvic acid's fate is determined by if you have oxygen present or not. If you have oxygen present, you go through the aerobic pathways, the citric acid cycle or Krebs cycle, and the electron transport chain, chemiosmosis, and you produce a whole bunch of ATP that way. If you don't have oxygen available, you go through the anaerobic pathway, and pyruvic acid is converted to lactic acid. Lactic acid can then go into your blood, and lactic acid can ultimately be converted back to glucose. And so your body has a remarkable way of turning anything that it has into anything that it needs. Here's a summary of the actions of glucagon, which will be the main hormone responsible for regulating this post-absorptive state. So if you have a decrease in your plasma glucose or rising amino acid levels, blood sugar is dropping, you need to make sure that it stays up. This stimulates the alpha cells of your pancreas to secrete the hormone glucagon. Glucagon tells your liver to break apart your glycogenolysis, and create glucose from non-carbohydrate sources, gluconeogenesis, and so those actions will help to increase your plasma glucose. If your glucagon will also stimulate your adipose tissue to release fatty acids into your plasma, and then you can use that fat and convert that into glucose if you need. As your blood glucose rises due to these processes, eventually your glucose levels will return back to normal, and then as you have normal glucose levels, this will turn off the secretion of glucagon. And so glucagon is regulated through negative feedback. As your glucose comes back to normal, you won't be secreting that glucagon anymore.